Good afternoon. For the next few minutes, I'm going to discuss one of the most powerful enabling technologies, gene therapy. Gene therapy can be defined as the transfer of a normal functional copy of a gene into a cell whose own copy of that gene is mutated. While essentially any disease could be treated with gene therapy, those caused by a single faulty gene are the most amenable, as only a single defect would need to be fixed. Some candidate diseases include the globinopathies, so sickle cell and thalassemias, lysosomal storage diseases, and a family of diseases on which I'll focus my attention, the hemophilias. Now, gene therapy can promise a precise and permanent cure for any disease that's caused by a genetic defect. This is in marked contrast to other current therapies, which are often inadequate and cannot promise to cure the disease. So to highlight the vast potential of gene therapy, I would like to spend a few moments using the hemophilias as an example. The hemophilias are X-linked bleeding disorders that were first described by Abulcasis all the way back in 980 AD, before people even knew the body was made up of cells. Being fairly rare, hemophilia didn't receive a lot of attention until Queen Victoria made it the royal disease. So since hemophilia is X-linked, women can be carriers of the mutation, but are phenotypically normal. Queen Victoria, without knowing it, was a carrier, and by following the timeless tradition of marrying her children, many of whom were females, to the children of other European royal families, she unknowingly spread hemophilia to several of the royal families, thereby affecting the whole continent's political future. Hemophilia A is the most common form of hemophilia and is an ideal disease to treat with gene therapy for several reasons. First, it's caused by a defect in only a single gene, the gene for coagulation factor 8. Secondly, expression of factor 8 need not be tissue or cell specific because the protein is secreted into the bloodstream. Importantly, very little of factor 8 is needed to produce a great therapeutic improvement. Also, supraphysiologic levels are well tolerated, providing a wide therapeutic window. Patients can present with one of three different phenotypes based upon the amount of circulating factor 8 activity in their plasma. Patients with less than 1% have a severe life-threatening phenotype that's characterized by frequent spontaneous bleeds. With just 2 to 5% of the normal levels of factor 8, patients present with a moderate phenotype, only rarely experiencing a spontaneous bleed, but still bleeding in response to minor trauma. At levels above 5% of normal, patients are clinically healthy, only bleeding in response to major trauma or surgery. As such, you can see why, if gene therapy could provide only 5% of the normal level of factor 8, this would create a marked therapeutic benefit and tremendously improve the patient's quality of life. Granted, there are currently treatments for hemophilia A, and these consist of injections of factor 8 protein. Unfortunately, these are very expensive, so ranging from about $250,000 to $1 million a year. A couple of years ago, a teen in Iowa was reported to spend roughly $1 million a month for his treatment. This led three of the major insurance companies to leave the Iowa market. Despite this cost and the fact that you have to infuse yourself usually 3 times a week, this is lifelong treatment without a cure. It's only available to about 30% of the world's hemophiliacs, and even among those who are fortunate enough to have this treatment, 25 to 30% of the patients will go on to develop inhibitory antibodies to factor 8. Now, to give you an idea of the impact gene therapy could have in the context of hemophilia A, using gene therapy to cure hemophilia A in just the patients born this year in the US alone would save the patients and the healthcare system about $4.8 billion. Grasa and I have been working for the past 15 years or so on developing gene and cell-based therapies for hemophilia A, using a unique line of sheep we recreated with several severe hemophilia A. And this model accurately recapitulates the human disease. Grasa is going to be speaking with you about that in more detail in one of the other sessions, so I'll focus the remainder of my talk on gene therapy as a tool. So how do we actually accomplish gene therapy? There are 3 ways we can transfer an exogenous gene into a desired cell type. The first would be ex vivo, and the simplest, in which we would remove the cells that we want to modify from the body, incubate them with vector, and then return the gene-engineered cells to the body. A slightly more sophisticated method would be in situ, in which we would take the gene delivery vector and place it directly into the affected tissues. And the final and kind of ultimate goal of gene therapy is to use it like any other treatment, and it would be in vivo, so we could take the vector and either administer it as a pill or directly inject it into the bloodstream. So having now seen the word vector several times in the preceding slides, what exactly is a vector? This is basically a delivery vehicle for transferring the desired genetic material into the target cell. This slide lists some of the more common vectors being used in research and clinically for gene therapy. And you can see this consists of pretty much any virus you can imagine, as well as some more sophisticated non-viral vectors that biomedical engineers are working on developing. To choose the appropriate vector for a given situation, we need to consider several different things. First, what are the target cells that we'd like to modify? Second, how much gene expression is needed? In the case of hemophilia A, we only need about 5%. If we want to treat something like sickle cell, we would need a much higher percentage of cells expressing the normal beta globin in order to get a correction. Are the target cells dividing? Do we need to have persistent gene transfer? Do we want to have an inflammatory reaction to the vector? For instance, if we're using gene delivery to try to remove a tumor, we would like the vector to stimulate an inflammatory response so the immune system comes in and eliminates the tumor. Would we like the option of being able to go back and repeat the transfer if it's needed? And how large is the transgene? In the interest of time, I'll focus on one of the most popular vector systems, the retroviruses. On the left, I've provided some pictures to help you recognize a retrovirus if you happen to see one. With all joking aside, the retro is actually called this because the genome is RNA, and this is reverse transcribed into DNA as part of the life cycle of the virus. Retroviruses and retrotransposons appear to be part of every cell's genome, so everything from bacteria all the way up to humans. Amazingly, about 8% of the human genome is comprised of retroviruses or retrotransposons. Importantly, these were the first infectious agents that were ever implicated in tumors, and these were chicken sarcomas identified by Rouse back in 1906. Now, why would we want to use a retrovirus as a vector? Well, retroviruses have evolved over millennia to ensure efficient entry into target cells. They have a wide host range, and we can modify the tropism fairly easily. They stably integrate into the host cell genome, which is a unique feature of this family of viruses. This enables them to mediate long-term expression of the transgene. And importantly, the genome can easily be modified for use as a vector. At least looking initially at the murine retroviruses, since they were isolated from mice, these were thought to be non-pathogenic in humans. Now, obviously, for gene therapy, we don't want to use a wild-type infectious retrovirus. Instead, scientists have developed ways of producing retroviral vectors that have been stripped of all of the viral genes and now encode only the desired therapeutic gene sequence. Moreover, these vectors are also replication defective, adding an additional degree of safety. As you can see in this slide, retroviral vectors are extremely popular and they are very effective. So as of yesterday, there were over 21,000 publications on PubMed using retroviral vectors, and about 300 of these are clinical trials. Going to clinicaltrials.gov, we can see that there are currently 45 ongoing clinical trials that are using retroviral vectors. While they are very effective, retroviral vectors aren't without their flaws. First, the capacity for the therapeutic gene is fairly small, so we can only package things that are less than 8 kb into the genome and have a functional vector. Infection is limited to cells that are expressing the receptor. And in the case of murine retroviruses, infectivity is limited to dividing cells. We can only produce relatively low titers, so about 1 million particles per mil, which is important if we're trying to treat a large adult that weighs 200 pounds. And the retroviruses are thought to, quote unquote, randomly integrate into the genome. And this poses two different problems. First, we can't predict the level of transcription. And secondly, it raises the possibility of insertional mutagenesis, which was considered for many years to be a theoretical risk. Unfortunately, the gene therapy community found out the hard way that insertional mutagenesis wasn't only a theoretical risk, but one that could actually occur and have very serious consequences. So back in 2002, one of the first successful gene therapy trials in the world for SCID was halted in France and the United States after a child developed leukemia following this gene therapy treatment. Subsequently, 2 more children in the trial later developed leukemia, and sequencing revealed that integration is certainly not random. Now, most retroviruses can only infect dividing cells, which eliminates some of the most desirable target cells, for instance, hematopoietic stem cells, muscle cells, or neurons. To overcome this obstacle, we first needed to ask why this limitation exists. And the reason for this is that murine retroviruses don't possess a protein that allows active uptake into the nucleus. So what could we do about this? Well, we could try to make a vector from a virus that has such a protein. And Luigi Naldini did just this and basically turned the world on its ear in 1996 when he showed it was possible to modify HIV and use that as a gene delivery vector and achieve efficient delivery into non-dividing cells both in vitro and in vivo. Amazingly, the first clinical trial using an HIV-based vector was conducted in 2005. Currently, there are 69 clinical trials using HIV or lentiviral vectors. Not surprisingly, HIV has the same disadvantages as the other retroviruses. In addition, however, I'd like to draw your attention to the fact that HIV is in fact a human pathogen, and one for which we don't have a cure. Now, up to this point, we've only discussed gene therapy in the context of simple gene addition. What if there were a way for us to actually correct the sequence of the genome and thereby cure disease? This technology is known as genome editing, and it's one of the hottest areas currently in biotechnology. Genome editing was born with the discovery of zinc finger proteins in frogs. These zinc finger proteins act as transcription factors using their blue coiled fingers that you see here in this diagram to bind to specific DNA sequences. Scientists quickly realized that zinc finger proteins could be engineered to retain their DNA binding fingers, seen here on the left and the right, but also encode a nuclease. Such zinc finger nucleases represent highly specific molecular scissors that enable us to introduce a double-strand break in any desired genomic locus. The cell then repairs this break using non-homologous end joining, thereby disrupting the function of the gene in question. Zinc finger nucleases are thus an amazing tool for creating knockout animals. Gene therapists, however, are trying to fix genes, not disrupt genes. So why would these be of interest to us? Well, if you introduce a repair template piece of DNA along with the zinc finger nuclease, you can get very efficient homologous recombination, in which the cell's machinery swaps the donor DNA sequence for the one the zinc finger nuclease has cut out of the genome. In this picture, you can see how the presence of a donor template DNA molecule enables the zinc finger nuclease and homologous recombination to swap an exogenous DNA segment in this darker blue color into a specific genomic site that's been cut by the zinc finger nuclease. Unfortunately, this technology isn't perfect. It's very complicated to design effective zinc finger nucleases. They're quite hit and miss and somewhat context dependent. Off-target effects can cause substantial toxicity, with each finger targeting only 3 nucleotides. One other issue that's arisen is that the company that first developed zinc finger nucleases, Sangamo, actually owns all of the IP associated with zinc finger nucleases and the algorithms to design them, which led to a publication a few years ago in Nature Biotechnology discussing the conflict between open science and the role of patent disclosure, and whether this was actually appropriate to kind of hinder the entire field from moving forward by having IP protection. Luckily for us, there are other options. So talons, for example, are also fusion proteins consisting of DNA binding motifs and the FOC1 nuclease. These were originally identified in plant bacterial pathogens, and they activate genes in infected cells to support the bacteria's growth. Talons are much easier to design and manufacture, as a simple cipher was developed that enables the researcher to determine the precise amino acids needed to target a specific sequence of DNA. Talons also have some shortcomings, sadly, however. They're very large, they're about 1,200 amino acids, so it turns out to be hard to deliver these to cells with high efficiency. Interestingly, because they have regions of homology, you can't actually use them in most viral vectors. They're bacterial in origin, so they may trigger an immune response in humans, and it's still unclear whether they're truly as site-specific as the software predicts. So in my last few slides, I'd like to take a few moments to discuss the latest and greatest in genome editing, the CRISPR-Cas system, for which Emmanuelle Charpentier and Jennifer Doudna just received the Nobel Prize. Now, all of the excitement about CRISPR-Cas is due largely to the fact that its DNA recognition portion relies on a guide RNA rather than a protein, and is thus determined by simple Watson-Crick binding. This machinery can easily be delivered to human cells. While it's still only a few years old, a PubMed search yesterday revealed over 24,000 publications using CRISPR-Cas, attesting to the promise of this technology. In this figure, we can see the CRISPR-Cas9 system in action, either disrupting a gene, which you can see with the arrow pointing to the top, or using homologous recombination to replace a genomic sequence with a donor template with the bottom arrow here in green. CRISPR-Cas has proven its value for generating not only small, but also large animal knockout models and large transgenic animals that express desired exogenous genes. The CRISPR-Cas system was quickly shown to be capable of mediating therapeutic correction in a lethal liver disease in mice, and to be able to ablate 2 key genes in human hematopoietic stem cells, a notoriously difficult cell to modify. The first of these genes serves as a co-receptor for HIV. By knocking out this gene, it rendered the HSC resistant to HIV infection. The second gene the investigators knocked out is a key part of the HLA molecule as a first step towards making universal donor cells that could be transplanted without matching and without fear of rejection. It's thus not surprising that leading journals in the field have heralded CRISPR-Cas as being poised to revolutionize both medicine and industry. So to close today, I would just like to acknowledge my partner in the lab and in life, Dr. Graça Almeida Porada, all of our lab members, and Dr. Atala and the whole team at W Firm. Thank you very much for your attention.